Good morning, everyone. We're going to start the last day and last session of this meeting. So it's just delay of 10 minutes because a lot of people were late this morning, including me. so now Hannezo will chair the first session. So we will have a break only at 10:40, and Ali Archok will be here to give the last lecture. So please stay until the end. He has some very amazing stuff that he wants to show you, and all of it is unpublished. So please stay all the way to the end. So now maybe we can have the first session.
Thank you, Alan, and welcome everyone. This morning we're going to have the reporting back of the breakout 3 sessions. And as you remember, there were 3 sessions, and we will basically do very much in the format as we did yesterday, which is about 10-15 minutes for each breakout group leads to report back, and then a discussion with the audience. And so for the first breakout group, I'd like to welcome Paolo Giacobini and Ines Sequeira, who led the discussion on health in later life, developmental origins of health outcomes over a lifespan.
Thank you, Maz. Good morning, everybody.
Hi, good morning.
Okay, so yeah, my name is Paolo Giacobini. I will try to report together with Ines the discussion that we had yesterday. And so the idea of this discussion was to actually raise some considerations and eventually awareness about an hypothesis that been proposed in the 90s by Professor David Barker. And the idea of these developmental origins of health and disease hypothesis is that perinatal exposure to adverse environmental cues may somehow predispose the development of adult non-communicable diseases. And this applies for several diseases in adult life, such as type 2 diabetes, obesity, infertility, and so on and so forth. So I thought it was a good thing to start thinking about, because we basically, as a community of HCA, have been focusing a lot on pathologies in terms of genetic pathologies, rare diseases, and somehow, for limitations that we'll be talking about it later, these kind of studies have not been really approached, and we're trying to see whether it's going to be feasible in the future. So let's say that the context here is that there have been a wealth of studies in preclinical models, and there are even epidemiological studies in large cohorts of patients, for instance, with autism, in which correlation between prenatal exposure to androgens, for instance, correlates very tightly with development of autist-like disorders, spectrum disorders. But globally, the idea is that maternal exposure to, for instance, enhanced metabolic input, such as nutrient status or hyperandrogenism, gestational diabetes, exposure to endocrine disrupting chemicals can all have an impact. And these actually predispose the fetus in an adverse womb to develop some rewiring in terms of gene expression or connectivity in different organs. So we basically had several questions. We tried to summarize 2 questions and swap with Ines back and forth about this if you want, or you can get just after this question. So the thing is that in order to use current transcriptomic technologies to address this question, we have a major issue of whether we can get these metadata information of the donors in order to actually track back this kind of questions.
So, hello, can you hear me? Good. So basically, we wanted to discuss whether if we want to study how, what's the impact of the environment, what's the impact of the donor's metabolic status in gene expression of the fetus samples, if we can have information regarding the method, well, if we can have extra information regarding the donor. And so we need to think about what is the data that we should collect, and then what is the data that we can actually collect, because that's one of the main limitations here. So we should consider expanding that metadata, and think also about how can we increase the impact of our studies, because we want to make sure that our studies live beyond, our data sets live beyond the publication, and that can be reused by other researchers asking completely different biological questions. We should refine the way we acquire the data and consider the resources that we are using for it. So ultimately, we have to start thinking about how can we make sure that the data that we are collecting now can actually be used in the future, and make it easily accessible, the metadata easily accessible for future studies, asking questions on, for example, diseases that Paolo was just mentioning. Can you go to the next slide? Thank you. So in terms of metadata that could be collected, one possible simple thing would be to look into the donor's BMI. So can we collect their metabolic status, their age, because we can correlate that with, for example, diabetes in the development of diabetes in the fetum. We can look into the gestational diabetes of the donor, the hyperglycemia status, and another very easy information will be to look at the medication. And one very interesting point that was raised is that sometimes you could easily collect amniotic fluid, and maybe we would be able to have access to this fluid and do some testing to see the levels of drugs in the environment and hormones as well. We will probably be able to access blood sample from the donor and to measure acute stress signaling, looking into hormonal balance, looking at the nutritional status of the donor. And there's more and more evidence that autism is associated with different level, well, with an increased level of testosterone. So if we can have that information from the environment where the fetus is, that will be very helpful to then do studies in the future when we have much more data, because of course we wouldn't be able to do this kind of correlational studies just with 7 samples. But if we start thinking about collecting this information now, in maybe 5 years' time, 10 years' time, when we have access to more samples, the information of previous studies will be there, and we will give much more power to the studies that we'll be conducting. So we were looking at the different centers where we are collecting samples at the moment. And so there is this human development biology resource based in Newcastle and in UCL in London. Well, there is the information about the donor is not given to the researcher. However, if you request, they have that information available. So we do have to have a special request, and when you apply to use that resource, to have that information. But the information is there. So if we think now, when collecting the data, the samples, that we could collect other metadata, maybe it will be interesting to have access to it. Then BDLRL, that is the component of the HDBR in the US, they were mentioning that actually is the participants that fill out the questionnaire, and usually they don't respond to all the questions. So there are gaps in the information, and how would that be reliable? And can this, we were considering whether we can actually assess some of the information empirically from the tissue itself. Can we use, for example, placenta to measure some of these metadata, some of these parameters that we'll be interested in? Another thing is the gender of the fetuses. This is not consistently screened in all the centers. So if we could always have that information associated on the metadata, that's going to be a plus. And then, of course, one of our big interests in Human Cell Atlas is making sure that we cover all the diversity and making sure that we have the information about the ancestry background of our donors. Of course, depending on the center, depending on the country, that information may be available or not. So something that we discussed was maybe we can have a population analysis and be able to infer that from this analysis, but again, will be correlative. So if we are able to have that information as well, that will be amazing. And so, would you like me to continue?
Yeah, for the perspectives, just to wrap it out and then we can discuss further. So we thought that eventually HCA could put some recommendations about the metadata or how we want to move forward to possible impact of the data set. The idea of harmonizing globally the data across different centers. Would these donors' metadata be possible to associate with the donated samples in the near future? And there was actually a consideration from NIH that there is actually a program called the Phoenix Toolkit, validated protocols to combine data in studies, and this is with a final aim to harmonize and get more statistical power as well. And
yeah, so ultimately we just need to start changing the way we think about when we collect the metadata for our projects. And I think it's really important for us to start discussing it and put together a set of guidelines that will allow us to harmonize the data that we collect from the different centers. And with that, I think we'll finish and we'll be happy to discuss further with you later.
Thank you.
Thank you very much, Paolo and Ines. We now move to the reporting back of the next breakout session, which was on ethics and patient researchers' perspective. And I welcome Bartha Maria Knoppers from Montreal and Sarah Chan from Edinburgh to give the report back.
No? It's not working. That light's bright. Okay, so we had a very interesting discussion yesterday with the people who attended our session, and I thought I'd just bring back some of the key points from the discussion. I wanted, first of all, to draw your attention to the work of the Ethics Working Group, which Bartha heads up, and to point out the toolkit and the resources that they've assembled. So the issues that the group raised yesterday will be familiar, I'm sure, to many of you, so issues associated with things like consent, particularly those working on pediatric samples. How do we do consent, and how do we more broadly do governance? in that sort of context. So just to point out that the toolkit that is available on the website has a collection of resources that can help you address some of these common challenges that you'll all be facing. There is material there on regulation and governance of the use of tissue and samples in a variety of different countries. There are template forms that help to address issues such as consent for minors, the reporting back of results, the application of data protection law in children. So just to draw attention to those and point out that they are there for HCA researchers to use. We then explored some of the issues that people are facing in their current work around the Pediatric and Developmental Cell Atlas. And there's an interesting overlap here actually with the report back of the last group who've raised some issues around things like the reuse of data and samples and also metadata. So one of the questions obviously is when we have tissues or samples or data that's been collected, what else can be done with that? And here the entanglement of the clinical and the research context presents particular problems, as again I'm sure many of you are aware. So where we've collected data or samples in a particular clinical context, perhaps around a given disease, what research use can we then make of those samples, and does it depend on what was said, done, agreed to at the time those samples were taken? So we discussed the example of newborn blood spots in some detail as a very common procedure in many countries, which leads to potentially having what could be seen as a biobank of biological samples that can then potentially, but only potentially, be used for research. So for example, if we wanted to do whole genome screening on blood spots, what would be the governance mechanism surrounding that? Who would then have access to the data? And so within this example, we also discussed the way in which newborn screening can be a pathway to ensuring access to social and healthcare more broadly. So it's a very common procedure in many countries, but also may serve as a particular child's entry into the health and social care pathway. We talked about questions around access to data. So where samples are collected as part of a public health program through the public health system, if researchers then want to access those, first of all, who controls that access? If data is generated, so if genome sequencing data or genomic data is generated from that, who would then control that? Is it the particular researchers who've done the data extraction, or is it the state who runs the health service? And we discussed the need for a more holistic approach to what Bartha called stewardship of samples and data. So that we need to look not only at who has access and who has control, but at ways of ensuring things like the quality of data, the quality of samples. And so here the approach to standardized metadata that we've been talking about here. How do we make sure that samples that we might be collecting at different times, across different populations, through different health systems, can nevertheless be joined up in a useful way so that we can make connections across those different populations? We discussed particular issues around things like anonymization. So often we might say that when a sample or when data has been sufficiently anonymized, the issue of individual consent to research use might become less important. And of course, the problem with genomic data is that you can never fully anonymize it. So your genomic data is in some ways uniquely personal, as we all know. So there's that tension between the more the identifying characteristics are stripped from a data set, the less use that data set can be. So if you think about taking out all of those characteristics about the individual patients that we might want to link up, disease state, environment, background, all of those sorts of things, the more we take those identifying or potentially identifying features away, the less use that data can be. So anonymization or de-identification isn't necessarily the ethical answer. And some interesting points were brought up about about needing to be able to identify different populations within our data sets. For example, to be able to look at health inequalities that might be the result of things like racial or ethnic background, or that might be the result of particular geographical distribution. So in other words, we need data to remain linked in certain ways for the public benefit of the research to be realized. And we talked quite a lot about different models of data protection. Bartha pointed out that our approach to protecting research data has been very much in some ways like the economic model of credit card protection, and actually that's not really suitable for the sorts of uses that we want to make of this biomedical data. So we then talked about issues with the reuse of samples. So if you have obtained consent for a particular piece of research or in a particular way, what happens if we then want to reuse samples? And so this is not even the problem necessarily with the clinical and research overlap, but if you may have obtained consent for research use to one particular study or set of studies, what happens down the line? So we have all of these potential resources, but if we can't use them because of changing circumstances, what happens to those? And we know that there are different mechanisms that have been proposed for dealing with this, so mechanisms such as broad consent, to say not just to this particular study, but to all forms of research use. But we have a problem, of course, with how broad is broad consent, and how broad can it be? So the point was raised that we're approaching 25 years since the first human embryonic stem cell lines were derived, and consent was obtained to the use of the embryos in research to generate cell lines at that time. But some of the things that we can now do with stem cells, so synthetic embryos or gastroloids, the use of in vitro gametogenesis, so making sperm and eggs out of stem cells, human-animal chimeras, brain organoids, these are all things that can now potentially be done with stem cells that were only imagined if that at the time that those cells were generated. So do we need, there's a sort of problem and a meta problem here, do we need fresh consent? Do we need some way of saying, well, these are forms of research that we didn't think about at the time, and therefore we need a new way? Do we go back 20 years later and seek consent? As we know, the problem with re-consent, particularly across population data sets and biobanks, is if we assume that no reply means no consent, you're going to get attrition of that set of samples or data. And that in itself is a problem for a reason that I'll come to shortly. So we need both to know what are the governance mechanisms under which potentially new forms of research can and should be carried out with historical samples. But we also need a way of identifying which kinds of research are the kinds of research that we need new governance mechanisms for. So there are those two sorts of issues. Now, I said that attrition of population data sets and biobanks is a problem, and it's not just a practical problem for research, that you can't do your research if you don't have access to the samples, but it's also an ethical problem. So I want to put this within a wider framework for thinking about research and ethics. So Bartha spoke yesterday about, particularly in this context, the UN Convention on the Rights of the Child, and the right to the highest attainable standard of health, which includes the need to carry out research in order to keep on improving the highest attainable standard of health. We need not just to think about the ethical costs of research that is done, so do we have consent, or if we're not using specific consent, what's the mechanism of governance, but we need to think about the opportunity costs of research not done. So research that can't be done, or research that isn't done as well as it could be, or research that is delayed because we don't have the governance mechanisms to make it happen, or because there are governance obstacles unnecessarily put in the way. That is an opportunity cost, and it's an opportunity cost that comes in terms of lives, in terms of children's well-being. So in other words, there is an ethical imperative to try to get research done, and therefore not to just assume that that attrition is just a thing that happens to our databases or our biobanks over time. So we need to think in terms of what the and I suppose, Bartha, you probably want to comment on this, what some of the upcoming tasks for the ethics working group, what are the priorities that we identified need to be thought about here? What are the future directions in terms of regulation and governance? Obviously, regulatory frameworks around the sorts of research that you want to do are in place, but are also constantly changing. And we spoke a bit about the impact, especially in the US, of the Dobbs decision. So Dobbs, of course, is most well known around the world for its impact on women's access to abortion and reproductive health services. But that's going to have a knock-on effect on things like availability of embryonic and fetal tissue, on the regulation of research with these tissues. So we need to keep an eye on what's going to happen around that and make sure that research, as far as we can ensure it, is enabled to go ahead. We need to think about these new forms of research that I spoke about and what sorts of governance mechanisms might be equipped to handle the sorts of research that we might think is maybe more controversial, that requires more public consultation and more input. And then we need also to think about new approaches to governance. So while consent absolutely remains important, we need to think about ways in which we might achieve governance of research involving historical samples, new uses of data sets, and that doesn't necessarily mean going back every time to every individual who contributed to the research to say, well, we're going to do something new, what do we do about it? So I think that's about that's my summary. Bartha, did you have anything that you wanted to add to that?
Well, you'll be hearing from me in the next session, so I'll keep it short. Just to tie into your first point and your last point, we had a bit of discussion about how newborn screening programs, which are screening programs to find the asymptomatic at-risk child, that these public health endeavors could be put at risk in terms of the best interest of the newborn if we were to start using the Guthrie cards as a research resource. And interesting from a populational point of view as that may be, if we move away from the presumed parental consent for newborn screening to requiring an explicit consent because we're doing whole genome sequencing, we could very well affect half of the world's newborns for whom the screening programs are their one time to be seen by a physician. So we have to think about the political long-term interests of the child in a political sense as well as in a scientific research sense. To get to the other issue, which ties into the previous presentation, when I saw all those questions about metadata and harmonization, and I thought interoperability, and I thought back to building biobanks, which we did a decade ago. And that's exactly the kind of discussion we had. How do we enter our data in every country where we have access to fetal tissues or to embryos or to umbilical cords, stem cells, etc., in a way that we can increase statistical significance by being able to share that data in a way across and around the world. And I was thinking what we should start working on is not going to see the women afterwards, but making it part of the prenatal workup to say, unless you opt out or unless you notify us that we cannot, we're doing this for umbilical cords in Canada, by the way, that we cannot use your tissue for protected, secure research, sometimes anonymized because there's no clinical going back to that person, it's simply to get the data. We will presume that your discarded materials can be used as a resource for the next woman, for the next child, and so on. And I'm not so sure that this won't appeal to a lot of people to think that another woman giving birth and another child being born and so on can actually be helped by building these shared resources in the future by making it part of standard hospital procedure, unless there's an indication of someone not wishing to participate in such an endeavor. So that's a dream, but the biobanks were built. There was a lot of protest. People didn't like the broad consent. They said it wasn't material, it wasn't specific. Well, it was specific. It was specific to building a broad, publicly available, protected, well-governed resource. How much more specific can you get? So it's broad, but it has its own goals. I think we need to think that way in the HCA as well, building some of those tools and building that, that drive towards public participation in resource building.
I've decided to use this mic. What will have a clap. So thank you very much, Sarah and Bartha, for the great discussion and of the breakout reporting on ethics and patients. And now we come to physics and mathematics of human development, which is quite difficult to study, and it would be really great to find out a bit more about how we can actually make more inroads. And I invite Edouard Hannezo and also Pierre-François Lenne to give the report back.
Okay, thank you very much. So together with Edouard, during this session, we try to highlight the importance of mechanical forces in shaping embryonic development, and we discuss the ways to integrate what we know about physical force with state of cells. And we framed we organized the discussion around 4 questions. One about the model systems which can be used to interrogate the mechanochemical feedbacks during development. Then we discussed the tools which are suited to image and as well to manipulate signaling as well as mechanical forces. And importantly, for the connection to this meeting, we discuss how modeling can be integrated to large data sets or mixed data sets. And finally, we had general discussions about what is a good model. Of course, it depends on the question, and we'll finish with that in a few minutes. So first, why do we need model systems to interrogate the mechanochemical feedbacks during development? And so the first thing we highlighted is that, okay, if you want to make an atlas, it's difficult because cells move, mix during development. Mechanical force drive movement of cells, but as well they drive fate specification. And because of these feedbacks, it's very hard to infer dynamics from static data, and we have to find ways to understand better the relationship between cell state, mechanical state, and for that we have different ways to tackle this question. We can look at mechanical signatures from transcriptomic atlas, and then nail down a few hypotheses that we can test in system in which we can look at the dynamic, and then looking at transcriptomic signature, how they change with forces using spatial assays, using organoids in different conditions. We can try to relate mechanical signature using transcriptomic data. It was proposed as well that we could use systematic CRISPR screens to understand better the connection between shapes, morphogenesis, and the mechanical signatures, and then we can again nail down to a few hundreds of candidate genes and look at specific systems. A question which was raised during this session, but as well during the sessions on organoids, was about how in vitro systems, organoids in particular, are compared to in vivo situation. This question can be tackled by transcriptomics as well, by looking at cell behaviors in organoids, and to some extent looking at transcriptomic data in vivo. So the next question we discussed was about the methods, which the tools which are the best suited to image and manipulate signaling as well as and forces. It's key to have live information, and for that the organoid systems are very suitable. We need optogenetic tools to manipulate force, and some are already available and can be used to force, for example, change of tissue shapes. And we can relate this manipulation to transcriptomic analysis. There are as well new force probes and new sensors which are compatible with live imaging and which allow today to look at instantaneous response. But it's as well important to understand the history of the cells in terms of mechanics, and for that we need to develop new tools. We think it's very important to understand, if we want to understand the feedbacks between mechanic and cell state, phase specification, we have to find ways, and there are already ways, to manipulate tissue geometry, for example, curvature, size, to understand better these feedbacks. And finally, we discuss as well tools which are already available, which are based on imaging and not on static images and not on dynamic data, which allow already from images here from epithelia to get to infer forces and some mechanical properties such as pressure and tension at cell contacts from data, from static data, without the need of dynamic data. And with that, I hand over to Edouard.
Maybe I go.
Yes.
Maybe I go. So then, I mean, we had a third discussion essentially, which is related, of course, to the point 2 that we just talked about, which is sort of concretely how do we integrate insights from physical and mathematical models with large-scale omics dataset. The first thing is we could ask, okay, why isn't it done? Why wasn't it done already? And I think it's largely because, in a way, I mean, the fields have been developing rapidly, both on the for instance, on spatial transcriptomics, but also on the field of sort of cell mechanics. In the past 10 years, the field of cell mechanics has mainly concentrated on understanding simplified tissues, for instance, tissues made of single cell types, but we've seen and we discussed in a number of sessions that a lot of interesting cases are when there's sort of gradients, temporal dynamics, mixtures between complex cells in 3D. And so for this, there's really a need to add complexity to these models, and in a way sort of merging this kind of physical modeling with omics datasets, both spatial and non-spatial, would be quite ideal. And so again, we had quite a bit of discussion on what should we measure in omics data. You know, is there an ontology that would tell us something interesting about the dynamics and the mechanical properties of tissues? It's a bit tricky because often there was a lot of discussion. Nowadays, if you try to do this, you get genes always the same, sort of like actin or like myosin, which are not extremely specific and don't necessarily tell you very much about the detail because for instance, mechanical forces are often fine-tuned by a number of regulators. But then there was a lot of discussion on whether we should maybe start with simplified cases where it might be easier to look at this. Again, we were discussing cases, for instance, where there's a long range gradient in the tissue, which we know, so where there's not, it might be a bit easier to look at sort of global properties. We also discussed, for instance, on boundaries between tissues. When we know that there's a boundary between tissues of different cell types, maybe that's where we could sort of interrogate sort of mechanical signatures of this. There's a big question that we had on at which scale should we look at this kind of data, because, for instance, what we discussed here, for instance, these ways of mechanically inferring forces from images of tissues, it only requires cell outlines, but this gives you some subcellular properties, which are, for instance, tensions, which are not necessarily trivial to integrate with spatial transcriptomic data, which can give sort of slightly larger scales. But we actually discussed that this might be a strength, because typically, if you try to infer mechanical properties at the subcellular level, they're quite noisy. But if you average over groups of sort of 5 to 10 cells, let's say 100 micron, which is something quite relevant for spatial transcriptomics, many of these sort of mechanical properties become very reproducible. And so maybe for some of these cases, it's not actually single cells that are more meaningful, but maybe actually sort of groups of tens of of a few cells, and this is the kind of places where we can actually look at trying to integrate these kinds of data sets. Again, these kind of inference, for instance, they're done on very simple tissues, epithelial tissues with no ECM, often in 2D actually, although 3D is continuing. So this is going to require a lot more sort of theories and data integration. So this was these sort of first and third point actually were mainly on how to integrate sort of physical forces with this cell classes. We also had quite a bit of discussion more on sort of models, let's say, of fate choices. So more the mathematical part rather than the physical part of the modeling. And the general question that we had is, can we really make use of simplified models, for instance, considering a few transcription factors or a few variables? Can we get from simple models fate choices, right? Something, can we predict something interesting and surprising about how a cell type bifurcates into two? Often the room was a bit skeptical that this kind of simplified models could work, because often they miss just a lot of relevant variables. However, one point that was made is that the whole point of how, for instance, transcriptomic data is dealt with is to use dimensionality reduction, and dimensionality reduction might be very good also for understanding a small number of reduced variables. With which then you could start doing modeling, right? Not necessarily the genes that you were expecting from the start, but some reduced variables that arise from the dimensionality reduction. And there were some ideas, for instance, that typically if you integrate around sort of 10-ish sort of variables from the dimensionality reduction, this would start to be quite, you could start making some relatively good sort of predictions, for instance, on the dynamics of fate choices. But of course, and it's going to be maybe the last point, there's a big question of interpretability, right? Once we have this model with these 10 reduced variables, these reduced variables are the sum combinations of very complex things, which is not necessarily really easy to relate back to something you would like to manipulate, for instance, through perturbation studies. And so the last point was maybe a bit more philosophical, but also I think useful to discuss when we talk about theoretical modeling, what's a good model, what is something you agree on. And this is something that actually people from very different communities have completely different definition of what is a theoretical model and what's a good theoretical model, right? In a bit of a caricature, one can sort of, at the two extreme, you have sort of physics models which try to be as simple as possible, but of course can miss a lot of variables and be less quantitative. And you have sort of models which sort of try to really learn directly from the data, machine learning types of models, which of course incorporate a lot more features of the data, but often have issues about interpretability. And so judging a good model is actually not sort of so obvious, because many people have different criteria, right? You could say that a good model is something as simple as possible, something that is derived from first principles, for instance. But of course, you could also say that it doesn't matter that you're simple and derived from first principle. What you want is to be quantitative or to predict something truly surprising about your data, no matter what the complexity. But of course, there are some interesting statistical aspects to this, right? The experimental validation of the model, should match the complexity of this model. And of course, surprise in a prediction is sometimes hard to define. But of course, I mean, a lot of the discussions were about the fact that we probably need to integrate these different ideas from different fields. And the last point we had is we often talk in development about bridging scales, right? So development is a clearly multi-scale process from molecules to cells to embryos. And we often talk about models that sort of completely bridge scale, being completely quantitative. We discussed that this might be quite unrealistic, because this is usually impossible, even for instance in physics. but it might be more important to have different models at different scales that make different predictions based on what you have, and on which specific, where you can still make some connections, not something really systematic, but where you can make some connections, because ultimately, of course, you want to relate specific molecular to phenotype relationship, and so to make different models at different scales communicate on some specific questions that are really important for specific perturbations. And yeah, that was basically the main discussions.
Thank you very much, Edouard and Pierre-François. So I think I'll actually please stay on, because I think all of us, all the 6 breakout reporters who led the session will come up here for any sort of any discussions. We've got a few minutes left. So if there are any points that anyone from the audience would like to raise, that would be great. I might kick off with one, and that's to relate the first and the second breakout. And I think, Bartha, you alluded to the fact that, you know, biobanks were built and it was possible. And I guess to some extent, you know, there are specific issues for pediatric samples and there are specific issues for developmental samples. And it's very rare to get developmental samples, and it's even harder to get some of the very basic information in some biobanks. And I guess for us to have a representative and inclusive developmental atlas, what do you think could be a way forward? Because they're very limited repositories, and some repositories don't give information even such as ancestry. Sex, we can easily infer from the data itself by looking at genes that are related to male and female sex. But I just wondered whether, is the solution to have more biobanks elsewhere, or is the solution to kind of like standardize some of the information that's being collected by existing biobanks to allow this information to be gathered?
I think the ideal would be to have infrastructure science, which is what I call biobanking and the study of tissues and data. Infrastructure science recognized as fundamental to medical care. And you have that feedback loop. And if people, citizens and politicians and so on, ministers of health, could understand that the quality of patient care is dependent on continuous refinement. What were the 3 R's I liked at the beginning? Reuse, refine, resource, was it, in the first slide? And I think that says it all. It's a simple concept. If we can get that through, that by going back, being able to understand the data for the next patient, that this should be standard medical practice. Now, this is a big leap, but I think it is possible. There are especially if it's like a pre-entry to the hospital. So it's not your specific research project that matters. What matters is that as a community, a developmental and pediatric community, you say we cannot serve our population in the absence of having quality infrastructure science. And then if you build these biobanks wherever they are, you don't need a big one like the UK Biobank, but you make sure that they're interoperable, that your variables are entered in the same way in a standardized, or at least not necessarily always standardized, but interoperable. I think you can make a real mark in the pediatric community.
Thank you. Are there any additional points? Yeah. We might need mic runners behind you and in front of you.
I have a question about the ethics and the reuse of historical material, because we have encountered this problem with, of course, patient material. And I wonder whether for the future, when we hope to have a broad consent, if this is feasible, first probably some lines should be prepared and given as guidelines, but I wonder whether we might need the help of people who work with law, because, for example, one possibility would be to get broad consent for all present technologies and future technologies, or people could say, I disagree to have genomic data or something like that. But then how to, I mean, the point is that what was rising before, if the technology changed and at the time of the biopsy was not even existing, the technology, this material will be blocked normally to be used unless there is a formula that let it use. So I wonder how this formula could be formulated, and if in addition to science we could have also some law people who could help in defining this, because it's not just a scientific problem.
So if the question is about for a specific context, or which I think it is, a specific procedure or a specific sample collection and following research protocol, there are well-defined ways. I mentioned the toolkit and templates on the website. So I want to draw attention, I guess, to there's research to be done in the ethics sphere as well. So those questions I raised about how do we reinvent governance for the new forms of research that are coming on the horizon, how do we deal with the complexities of whether or not consent is required, those are ethical research questions to which we hope to develop answers in the coming years, in terms of what should you as a researcher do around your current consent protocols and how you go about collecting samples. There will be well-defined institutional, or there should be well-defined institutional procedures. There are best practice guidelines. So in terms of what to do right now today, go to those resources and follow those, and your hospital ethics committees or your institutional ethics committees will also be able to advise. But then in terms of rethinking those ideas more broadly, I mean, I think what Bartha proposed about moving towards a more understanding research as an essential part of the healthcare ecosystem, I think as well, we've maybe become too focused on looking at things like individual consent. And when we go to publics and we say, what's your opinion on things like biobank research, often you actually get a very positive response, or you get people saying, I thought you were doing that already, why wouldn't you do that? You can do so much good with this stuff. So I think another part, I wouldn't say the other part, but another part of the puzzle for us also has to be going out and looking at those attitudes and learning from them and saying, actually, if people expect that we're doing the research, why aren't we doing the research? But for the specific question, procedures are in place and the resources are there.
I mean, I just echo that in terms of one of the public engagement event we did, One Cell at a Time, which is actually available. We had a thing about consent and getting feedback from the public, and it was actually quite amazing to hear how much they value the research and were surprised by how much things were done or not done, just to echo your point. So for anyone interested in that public engagement, do go and check out the website, onecellatime.org. I think we have time for one final question.
Yeah, I just wanted to make a comment on, and I think it's already been spoken about now in answering that previous question, but just the idea of if we're going to have like an idea that's a national sort of protocol that is sort of laid out at a government policy level that, like Martha spoke about, of integrating biobanking into just the general health process, I guess. It would be also interesting to see how that then filters down to the institutional ethics committees, because I think that, and it's something that we face at our committee, that even if the laws say one thing, it gets like more and more stringent as you get into the actual institutional ethics, and then often it's not actually the same, and you get limited by your institution, even if say there was a national policy that allowed you to do something, your institution doesn't want to take the risk, and so it doesn't get filtered down.
Yeah, oh, sorry.
Yeah, I think that there's one more consideration to take into account is that, yeah, I agree with the institutional ethics, but for instance, for us, it's more each country has its own ethical regulations, which is actually very hard to break because it's at the ministerial levels. As we were mentioning yesterday in our working group, we cannot get information about ethnicity or ancestries, and whereas in England it is possible. So one propositions that we had in the audience is that maybe we should do some harder lobbying toward the politicians and see for instance, with great examples of the community, saying this research is possible in this country, we're affiliated to this community. Can we move forward with the idea of sharing resources? Because at the end of the day, what we were proposing yesterday, and I'm glad there's been raised again today, is that we should move toward a concept of being able to share resources. Because we're talking about precious material, we can get a lot of information. I might be looking at one particular system, but people can use the same material I'm working on to interrogate other systems. And this is, I think, the challenge for the future, but it requires a lot of lobbying in terms of political interventions.
Is that, Sarah, do you want to make any last comments?
Yeah, I guess just in terms of, I guess if you want to call it an ethical paradigm shift in the way that we approach research here, at the moment we have an approach that is very much no research unless explicitly yes, and I think all of the work that we're doing suggests that with appropriate protections in place, we should be thinking a little bit more about yes unless no. And then the other way of thinking about it, we're very focused often on the individual, but these are not individual, it's not research at the individual level, it is the population and the populations that make this research possible. And so we should be moving away from it's my stuff, it's my say, to it's our science, and we collectively, publics, have more of a say. And how we translate that into the implementation, as you've pointed out, is an entirely different kettle of fish.
A profound statement. I think we should come to a close, and I'd like to thank very much our breakout leads and everyone here for the great discussion. Thank you. And now I welcome our next chair, Rosa Ventatomo from the Wellcome Sanger Institute.
Thank you very much. And after this nice discussion, we start with the plenary session 3. And it's a pleasure to invite Galabina Yordanova and Ida Zucchi, who will be discussing about the HCA data platform and how can we support and interact with it. Thank you.
Hi everyone, and thank you for having us today. Actually, our talk is quite relevant to everything that was discussed so far. Obviously, we're here to represent the data coordination platform and specifically the work that we're doing right now with the bionetworks. Yeah, we want to focus a little bit on how we are supporting at the moment, like different levels of access. But yeah, just an introduction. I'm Gabi Yordanova. I'm a product owner for the HCA data ingestion service as part of the coordination platform. And I'm here with my colleague Ida, who is actually a data curator. So Ida has already worked with quite a few of you here in the audience. So yeah, just a little bit about the platform for those people who don't know about it. And then we're going to just talk a little bit on how we're currently implementing the different policies of access and what we consider like suitable for data curation into the data platform. And then at the end, we're going to just briefly say what are the current steps that people can take in order to get in touch with us, and we help you curate the data and metadata for your projects. So yeah, very briefly, the Human Cell Atlas, everybody from this audience knows what it's about, but I want to focus that obviously there are hundreds of labs generating all kinds of data focused on their own research questions, their tissue of interest, and obviously using different experimental techniques and data types. And also we consider different user personas, the people who are actually going to otherwise make use of that data. And as Ines said, as just considering the people who are going to kind of try and use the data in its further life, not just, the life of the data from the publication, but there might be more people on the experimental side or computational side. At the same time, we're also dealing with metadata, which is considered, important for, let's say, one organ, let's say for lung smoking history. I'm not sure how the microphone is, but yeah, let's say smoking history is very important metadata for the lung community and for others. There is specific metadata that was being mentioned already when we are speaking about, let's say, developmental and some metadata attributes relevant to the mother or to the environment. And also, of course, we need to consider data with different types of access, whether it's completely open access or controlled access, which at the moment we consider all data from living donors as controlled access. So our question here is, how do we bring it all together? And we at the HCA Data Coordination Platform are trying to kind of be in touch with the community and make sure that all the single-cell data generated could actually be found, could be stored, and we can provide access to it. So we also, we follow the FAIR data principles. And again, that's a variation of reuse, refine, recycle, I think. But so we make sure where can the data be found, how to access and download it. Can it be interoperabilities about, can it be analyzed further by a machine or another researcher, and is it reusable? And yeah, the impacts that we're after is just, we're trying to be a comprehensive resource. We also are working, and we have an established like ATA metadata standard so that it best supports integration efforts and just like further interrogation of the data so that it supports like future insights. And we're also making sure that there are connections from the data on the portal to further analysis and visualization portals, such as, you know, there are multiple of those, but Cell by Gene or the Cell Browser. And I just wanted to briefly have a side note here, but let's say things that we saw from the breakout discussion. Ines raised the question about, let's say, things like BMI or other attributes. I just wanted to say the metadata standard that we have actually supports quite a lot of these attributes. We have them structured in a really neat way, but again, the problems in most cases come from, if it's living donors, we would actually encourage people really to gather all of this information, but at the moment, we would probably direct people with such metadata to actually go and deposit into a controlled access repository when it comes with living donors. Okay. Very briefly, how the DCP works is labs contribute single-cell data. This is where we come in because we specifically represent the EBI. We deal with the curation and ingestion of the data. And then data gets uploaded and indexed and stored. That's the Broad Institute's responsibility, and the University of Santa Cruz is responsible for the data portal, where basically all of our efforts into metadata curation and uploading of files, etc. So that's where it ends up. And obviously, the way we have curated the metadata on our end would result in different filters that you can find on the data portal. And every project is represented with you know, the files available as well as the metadata. I just wanted to briefly mention our work with the networks. Obviously, in the years so far, we have focused on curation of data that we have discovered either through events such as these or through publications, but ever since the Vienna meeting, actually in June, we saw the big focus and just the energy around the networks trying to compile the dataset lists for integration. So at the moment, we have decided to focus our efforts mainly in working with those groups. And how we're trying to help is, first of all, to make sure that all the data sets that the scientists who have chosen to be part of those integration atlases, we're trying to curate them as a priority. We have started working with these groups that you see on screen, but obviously, there is like a backlog. And we're trying to help them by obviously showing the metadata standard that we have, trying and actually identifying common data elements between the different metadata elements between the different data sets, and also trying to work around, working through issues that are around sharing. And on that subject, I think I will hand over now, after this brief introduction, to my colleague Ida, who is going to just focus a little bit more on very specific details on how we deal with data submission.
Hi everyone. I'll briefly go over to the stability criteria with an accent on access type. So we have 5 main criteria, the first one being the species. We mostly focus on human and mouse data. We can accept data from other organisms, and given the conversation we've been having, this might be particularly relevant. So if you have a data set that doesn't belong to human or mouse, you can get in touch with us and we evaluate data sets on a case-by-case basis. Then we accept sample types from organoids, primary data, primary tissue, and cell lines. We accept a wide range of technologies. You can see some examples on screen, but we're always working to support more technologies, and we're very keen to support all the work you're doing. We accept both samples from healthy tissues and disease donors, and this particularly means even just datasets that only include disease donors. This is a bit of a more recent development, so good to bring it to attention. And more on the access type. So if the dataset you have, it's completely open access, we are very happy to welcome raw sequence data, analysis files such as count cell matrices, cell type or spatial embeddings, and metadata on the donor and sample level. And we qualify as donor metadata things such as age, sex, gender, smoking or drinking history, medical history. And we consider sample metadata things such as the organ part where the sample was taken and the disease and health status of that specific sample. And things such as all the protocols that go into actually making an experiment, so dissociation of the tissue, enrichment, or any other things like organoid protocols, any information that we can keep on the experiment that was carried out. So in the case of a controlled access dataset, on the other hand, we try to keep in line with GDPR regulations. So we only accept analysis files and metadata on the sample level. And again, stress on the living donors case to be very cautious in protecting the potential to make sure that our donors are not exposed to re-identification. We actually treat anything from living donors as controlled access, so we only accept analysis files and metadata on the sample level. So how does submission work? Let's say you have a data set and you would like to submit it. The first thing would be getting in touch with the Wrangler team. I'm a member of the Wrangler team, and you can see our email on screen. If you get in touch, then we would walk you through a suitability criteria questionnaire so that we can assess where you are and what we can do to help you. You would have to sign the terms and conditions for the data to be published in the Human Cell Atlas data coordination platform. And then at that point, that's where we start to go back and forth so that we can get all the metadata that we need to accurately represent the experiment you've done, going from donor and sample information, protocols, anything that we can get. It's all structured in a flexible way. So once all the metadata has been curated, we do a sub-validation to make sure that the format that we've done and all the metadata we've gathered is it matches up with the schema that we use so that all the datasets are formatted in a consistent way, which makes them interoperable. So there's this validation step, and then there is data upload. So if your sequences, if your analysis matrices have already been archived somewhere, we can collect them on our own. But if not, we can give you instructions on how to upload them directly into our system. And in certain cases, we can even help with archiving to databases such as ENA. And once everything is done, we send it off to the data portal. So the submission is complete. At this point, if we've done archiving for you, you would get accessions back that you can put into papers and works to just have a reference to the data. And then you're acknowledged on the data portal as generators of the data. And at that point, the data and metadata is publicly accessible. And that is what powers essentially the search capabilities on the portal. You can search with a lot of different criteria, ranging from any kind of donor information, the tissue that was used, or file types. Maybe you're interested in a specific file type, or even free text field. So, as Gabby said, we also try to work with other initiatives. So we we curate analysis matrices so that it can be then visualized on the Cell by Gene portal or the UCSC single cell portal. And we also try to stay in contact with other initiatives such as the single cell portal, the single cell expression atlas, and the Cambridge Cell Atlas. One last note, we release data monthly. So if you go into the portal and you want to see what is new, you can check out and you look for the blue dot that will tell you what has been recently published. And lastly, we very much welcome feedback on anything going from how the portal looks, how the submission process works, or even just working with the data. And there's an email on screen again for that. There's a DCP community Slack if you want to join and see where the discussions are going. And lastly, again, you can see again our email on screen for the Wrangler team. So if you would like to submit your dataset, please get in touch. We'd be very happy to work with you. And this is the John Zuckerberg initiatives. Our main funders. These again are the people who actually work on the portal: the Broad Institute, the Santa Cruz University of California, and EBI. This is the actual team. So. Just so you can see them, they're all here. And again, these are other initiatives we stay in touch with and other funders. That's it. Thank you for listening.
Hi. Do you actually have data that are coming from labs in Europe, France, and others? Because, like, as Rosa knows, we have a European grant, and we were not allowed to put anything on the HCA portal because we were told that you were not compliant with the DPRs. So I mean, Rosa can testify. We were told that we cannot put anything on your portal, so we had to put everything on other solution. So EGA or things like this, but for now we were not allowed to put anything on the portal.
Okay, so yeah, this was partly kind of covered. We do actually curate data from Europe mainly as the EBI, while our colleagues from UCSC are curating data sets coming primarily from the US. When it comes to, again, if it's like live donors, just because of GDPR concerns, we would actually encourage people to put the raw sequencing data into the EGA, but we will always get the analysis files and put them onto the DCP.
Yeah, but for now they were like embryo data, so the embryos for sure they're not alive, and yet we were not able to put anything on the HCA portal. Oh. No data. So I mean, Rosa is aware of it. There was no way we can put something. So from the UK they can, from France, and so because it was European consortium, we couldn't put any data.
So
So we can discuss later, but for now, there's no solution. I mean, we went through various reviews, ethics, etc. And we were told you cannot put anything.
I think, yeah, I think probably email us and we can kind of follow up. But yeah, our policies were as the ones, yeah, on screen, which was we would actually count matrices we would be able to get. Yeah. Yeah. Yeah.
I was just wondering on that note about only being able to have count matrices for living samples or living donors. Well, I don't know how that getting a lot of feedback. How that's going to influence integration in the future, because I guess that's the whole point, is to be able to integrate with other data sets. But you're saying that you'd encourage us to put the fast queues in another portal. Is that? Yeah, can you explain more about that?
Yeah, fast queues we would encourage at the moment for them to go to like a controlled access repository. However, the HCA itself, there's just a lot of processes still going on in terms of like legal steps to be taken. But the HCA itself in the future is also going to support managed access, which means that, you know, those files potentially would be on the HCA data portal as well when it establishes its own like data access committee, etc. It's just, yeah, we encourage people to kind of for the moment use managed access repositories, but in the future we're going to make sure that, yeah, whenever we have managed access on the DCP, those would actually be there as well.
But so by managed access, do you mean somebody would have to like apply?
Oh, sorry.
Do you mean somebody would have to apply and get a password access and all of that? That would be how it would work.
That's the current situation. And again, we're following here just everything that is kind of recommended by the ethics and legal group. I'm sure in the next talk this will be covered as well. But yeah, the DCP at the moment is just following what the rules and regulations are, and we're kind of following on what are the what the ethics group is recommending.
Just to clarify, your particular point about the HCA not being compliant with the General Data Protection Regulation, a lot has happened since that original worry about the impact, including creating an HCA Inc. in Europe and an HCA Inc. in the United States, so that different regulations could cover European contributions to the data coordination platform. There was also a data access committee for the controlled access being created. So all those issues about controllers and processors and everything else that would make it compliant with European GDPR have been taken care of. So the original worries, including using the Microsoft or whatever cloud that is particular to Europe, because there were worries about China, the United States, et cetera, a lot of those legal issues have now been resolved. So some of the original hesitation, I think, could be dissipated by just finding out what the new rules are and how these new rules respect European regulations.
I think we have time for a very, very quick question with a quick response, please.
Hi. Oh, it's too loud. From Sweden, as I mentioned in my presentation, we are also working together with the National Bioinformatics Infrastructure to deposit and help them implement the local EGA. So this is actually just an ongoing effort across many countries. As of now, in Europe, Norway, Germany, Sweden, Finland, and Spain, I think, are the 5 countries that are, and UK, of course, are the ones allowed to deposit data in these local federated nodes. The other countries are still working on that. So many of the other European countries are also part of the Elixir, which is the life science data infrastructure in Europe. I mean, this is an ongoing effort. I mean, we have been trying to implement that for almost 4 years now after GDPR regulations. And that is valid for all the sensitive information that you have. So then that should be deposited in the country. The regulations say that the data that is produced in a country, let's say Sweden, should not leave Sweden, and that's why Sweden needs to have its own local EGA to protect data from being sent abroad or going into a server or to somewhere else that is not in Sweden. Just an addendum onto that.
For the detailed explanation.
Yeah, maybe we can discuss this later. Thank you very much. It was super useful, but now we need to give rise to the second speaker, who is Partha Majumder. And they will be talking about the commitment and activities of HCA to promote equity and some results from HCA-related research from India. Thank you very much.
Thank you very much. I represent, I'm a co-chair of the Equity Working Group, and I'm speaking on behalf of the entire Equity Group. My colleagues as co-chairs are Alex Shelley from Northeast. Oh, okay. All right. So, yeah, thank you very much. So like I said, that I'm representing the Equity Working Group, and along with me, there are two other co-chairs, Alex Shalek from MIT and Musa Malanga from Radboud University in Belgium. So essentially, we've been working on equity, and HCA from its very inception has been committed to promoting equity. Next slide. This is fantastic biology, building the human body one cell at a time, but eventually those of us who are interested in translation of all of this knowledge, essentially this is all a build-up towards more and more better and better precision medicine. But again, we've heard comments saying that genomics is a double-edged sword. If we use carelessly, genomics may exacerbate disparities, and that was in 2010. Subsequently, there have been also appeals that precision genomics need to be implemented primarily because precision genomics can actually promote health equity. There has been some penetration of precision genomics to significantly improve or promote health equity, but much more needs to be done. Minimally, the pace of penetration needs to increase. It's dependent on access to knowledge, dependent on access to technologies, and dependent on reduction of cost of use of these technologies. The HCA is trying to help in all of these aspects in terms of knowledge, access to knowledge, technologies, and also working with the manufacturers of these technologies to reduce costs, not just in the US or wherever the manufacturers are, but also throughout the world. Currently, HCA, many of us believe, is the pinnacle of precision genomics. It's defining human body cell by cell, as we all know. Eventually, the information will enable even better disease management. It will save on cost of treatment. Those are really the hopes. Reduce patient suffering, reduce anxiety of family members with respect to what's going to happen with respect to the treatment that's being provided to a member of the family who is suffering, increase patient survival. And all of this we expect will benefit as we understand the human body and the crosstalks between various human cells in the human body. It will benefit across geography, ethnicity, gender, and age. That's what HCA leadership believes, and that's what all of us who are contributing to HCA believe, that all of this really represents equity. There was an editorial in the American Journal of Respiratory and Critical Care Medicine, and that was 6 years ago. And essentially what it said is that it needs to consider social, environmental, and genetic factors to ensure that all individuals receive the right diagnosis and treatments at the right time. If it's conducted in a socially just manner, such that the rising tide of precision genomics lifts all boats, including groups disproportionately affected by disease. And as a matter of fact, because of the belief that equity needs to be promoted in HCA, the HCA has been doing various kinds of activities that's related to promoting equity. We've also know this, and it was in the same year that this paper was published as a commentary in Nature, that genomics is failing on diversity. And as a matter of fact, all of us have discussed, even in this meeting, that there is a disproportionate number of disproportionate data that's been generated, disproportionate amount of data that's been generated from people of European descent, and they may not represent all of humanity. There is, we recognize that there is considerable variation among populations, and we cannot ignore these variations. Therefore, there has to be better representation of various population groups and individuals from across geographies and other ethnicities to contribute to the data of HCA, such that the inferences drawn in HCA become generalizable and applicable to all of humanity. Inclusivity is a commitment of the HCA. To promote equity, HCA is actively taking actions to share knowledge of concepts and technologies, and this is the way that this is being done is to organize workshops and road shows, especially in low and middle income countries. It's empowering scientists from underserved and less endowed countries to participate. We are engaging them as partners in research and translation, providing access to technologies, and educating them, and in order to build capacity in those countries, and also be educated about the infrastructure, about the population diversity, etc., in those countries. Enabling provision of less expensive and more portable technologies and reagents, preferentially to LMICs. And one of those recent technologies is the ability to actually carry cells drawn from an individual and store them for a while before processing them for various kinds of assays, single-cell assays. The HCA working group has released several publications and statements on its goals and activities. Some of you may be aware of these. The Human Cell Atlas and Equity Lessons Learned. Essentially, it's a compendium or a narrative of 6 lessons learned that also includes identify, articulate, and track how equitable partnerships on multiple levels lead to long-term empowerment, mutual benefit, and equal participation. And these for each of these points, the description or the narratives are fairly long, and if you're interested, one should go back to that. The Human Cell Atlas supports Black Lives Matter and global ethnic equity. So essentially a statement and a document was put out in the public domain that, among other things, stated that we remain committed to the values of equity, collaboration, and human health for all, as well as to engaging, including listening to and amplifying the voices of scientists and research participants from marginalized communities. And indeed, that statement was taken well and was publicized by other groups as well. The commitment of Human Cell Atlas to humanity, this is again a major piece of writing that was initiated from that first narrative that we provided, and this is under review in one of the major international journals. We've also published a Nature correspondence recently in Sharing HCA Benefits Humanity, and in that one of the major statements that we made is that the consortium, the Human Cell Atlas as a consortium, strongly opposes exploitation of research results for any form of discrimination or racial profiling. Right now we have, this may be a week or two old, we have in our database 33.3 million cells taken from 4.6 donors. And these donors have come from many parts of the world. This is not to say that there is no skewness in the donors or in the cells that have accumulated, but again, the skewness in terms of geography or ethnicity is, one is acutely aware of this, and we are all trying to mend this such that it becomes really representative of all of humanity. Use of the data and potential for discrimination have been discussed, and biological data we know have sometimes been used for racial profiling and discrimination, although race is a social construct but and not a biological one. But we have, in the HCA, we have strongly opposed through various kinds of publications and holding road shows, etc. We strongly opposed the use of these data for biological differences for social discrimination. Some examples, early examples. This is in London. The initial equity strategy meeting group was held in 2019, followed by another roadshow that was held in Sao Paulo, Brazil, which was very widely attended and very successful. Subsequently, a huge meeting was held as a sort of back-to-back meeting with a major Gates Foundation meeting. This is the first HCA equity meeting that was held in 2019 in Addis, and several of you may have participated in that Addis meeting. The education and training remains a major focus of the HCA equity working group. I'm not going to give you a listing of all of this. Let me just tell you what has been done this year. The year started with a major workshop in India in the institute where I belong, and this was held jointly with the MIT. There were over. This was of course online. There were over 200 participants. The countries represented were Bangladesh, France, Germany, India, Pakistan, UK, and the US. Subsequently, there was another workshop that was held. This was also a virtual. It was a rerun of a workshop that was held in Africa from 17th to 20th October, and then a virtual rerun was made very recently, 14th to 16th of November this year. There are other meetings and workshops that are happening. This was held 3rd to 4th November in Bangkok, Thailand. This was a hybrid meeting, and this was in the HCA Asia meeting, but equity was well represented in that meeting. Coming up, Vietnam and the Middle East. Vietnam should have, the roadshow and engagement in Vietnam should have happened before the pandemic broke out, but because of the pandemic, it's been delayed. We are again back in talking with scientists in Vietnam, and again, many scientists in the Middle East have been contacted, and many of them have shown interest such that we hope that we'll be able to hold a major equity meeting in the Middle East recently. Not just the equity meeting, but also hands-on training, etc. The Human Cell Atlas Africa in-person meeting is going to happen the 7th to 10th March in 2023, and this will be primarily on computational and experimental design. So we are doing various kinds of things to promote equity. HCA health leaders are providing training. I myself, or many of my colleagues, have been direct beneficiaries of this attitude that HCA has towards promoting equity and facilitating training. I would like to express, so I have 2 or 3 slides to share with you as to what we have done subsequent to the training that we got at the Broad Institute, at the Wellcome Sanger Institute, and later in MIT. Aviv, Sarah, and Alex have really helped us, and we have been trained in these various institutes to initiate work in India in the single cell area. And we've been working on a disease. It's very difficult for us to, in countries like India, to generate money for HCA unless it's one talks about in the context of a disease. So we've been talking about in the context of oral cancer, and we've also been generating data from normal cells in the buccal mucosa or buccal cavity. And we've identified, this is also very pertinent to this particular meeting, that meeting which deals with developmental and pediatric programs. So we've been able to identify 2 major cellular programs. We are talking about oral cancer, which is the 8th most common cancer. About 250,000 new cases arise annually. Most of them, 2/3 of them in developing countries, about 125,000 deaths happen annually. Accounts for about 1/3 of all tobacco-related cancers in India. The site distribution of oral cancer is very interesting. In the West, it's predominantly tongue, and is associated with human papillomavirus. In India, it's predominantly, it's only about a quarter, which is tongue cancer, but predominantly cancer of the entire buccal cavity, which is called the gingival-buccal complex, minus the tongue. So there is a great anatomical contrast between where oral cancer happens in Indian patients and in Western patients. In India, it's primarily driven by tobacco chewing, or the environmental agent that's associated with oral cancer is primarily chewing of tobacco. In the West, it's primarily smoking of tobacco. What we've done is to do, again, single-cell work. I'm not going to get into the details of it, but essentially what we have found is that 40.61% of 40.61% of malignant cells express genes that are involved in EMT, and the HIF1 expression level positively correlates with expression of genes in partial EMT genes. So that's one identification that we have made, and we've, of course, identified the extent of diversity, et cetera, and I'm not going to present those results. The other is fetal and germinal epithelial cells are abundant in malignant cells. So it's essentially 40% of EMT, 40% of fetal and germline epithelial cells, and patients who are enriched for one of these two pathways, one of these two developmental pathways, are completely disjoint. So the same patient is not showing both of these developmental pathways. There are other detailed results that this is really, I'm not really giving a scientific talk, but there are other detailed results. The only reason why I'm presenting those is that we have been beneficiaries of the promotion of equity that was undertaken by HCA since its inception, and we've been able to contribute to HCA's scientific efforts, and also in particular to this particular meeting, our results actually dovetail very well to the theme of this particular meeting, primarily because fetal cellular reprogramming and EMT or partial EMT play major roles in this particular form of oral cancer tumorigenesis. These are really recapitulation of early developmental programs. So I would like to end here and say that we continue to promote equity and essentially to promote the technologies and the knowledge of HCA such that other underserved and developing nations can actually participate and contribute to HCA. So we will continue to do that, and this was a snapshot of what we have done in the Equity Working Group. Thank you very much for your attention.
Thank you. Any questions? So I will start just by asking a question on how can we what is the best thing that we can do to proactively help with the diversity of the data collection? Is just helping to have more funding, or what would be the best?
So again, as we are promoting HCA through these educational programs, etc., there are many scientists who are actually expressing deep interest in contributing to HCA, and we as Equity Working Group are trying to entice, let me use that word, scientists in in the underserved countries and less represented countries in the HCA, so that the diversity of cells in the HCA increases. So that's being actively thought through, and we are actually promoting HCA in those countries and bringing those scientists in. That's how we are doing it.
Yeah, that's fantastic. Thank you so much.
Thank you.
If we have time, I just have one question. Thank you very much. That was
a
fantastic session. I just wanted to ask one question about equity of access. So in the West, we generally enjoy quite a lot of bandwidth and a lot of compute power for the significant amount of data that we produce. So I was interested in your thoughts about how we make sure that everyone globally, if they might not have access to that kind of bandwidth and access to compute power, can still kind of answer the questions and leverage the full power of all of the data that's being generated by the HCA. Thank you.
Thank you. Again, like I said, that the easiest way to get in touch with us would be to get in touch with one of the co-chairs. And if, for example, you want to hold a workshop in your own country and you want help from the Equity Working Group, we would be very glad to come and participate or even generate funding to organize that. Right now, we do have funding to organize some workshops, etc. So very glad to do that.
Thank you very much. I think we need to move on to the next speaker, and thank you. So the next speaker is Bartha and Knoppers, and they will be talking about tracking ethical provenance. So thank you very much.
Thank you. I'll try not to keep you from your coffee for too long. Might be considered unethical and against human rights. So what I'm going to describe to you today is actually research. It's only been about 2 years now that I've been thinking about this issue. And it started not only because of experience with the Human Cell Atlas, but in particular discussions with the Ethics Working Group, and specifically in building the ethics toolkit, which I encourage you to check on in your computers and actually find the tools that have been built in the last 3-4 years by the Ethics Working Group. This is the wrong slides. The slide should be the ethical provenance slides that I sent in. I'll continue speaking to it while you find them. All right, so ethical provenance, what is it? That's the one from yesterday's breakout session. So they're the wrong slides. If you could check it out, Christine. Thank you. So what is ethical provenance? It's the practice of developing a record of the ethical, legal, and institutional rules applicable to data use for specific data sets, and it includes the permissions for the data. So what it actually does, it builds a toolkit that helps you find out in a transparent way what was the journey of the data from the original collection to its actual use by researchers, clinicians, and so on. How did we come about this? Well, the Global Alliance for Genomics and Health, which is an organization you've probably heard of. In fact, the Human Cell Atlas is a driver project in the Global Alliance for Genomics and Health, and it was founded in 2013. The aim of this GA4GH, as it's commonly called, was to accelerate progress in genomic science and human health by developing standards and framing policy for responsible genomic and health-related data sharing. So this was an idea that came sort of post the human genome map having been completed in 2003. And the reason it's important, because I can see with the HGA the same process of data sharing that is occurring. What's original about GA4GH, it's the first international organization that decided in the scientific community not to say we're going to follow this school of ethics, or we're going to follow the laws of this country, or we're going to see how we can get everyone to work together even though there are differences. It went to a different level. It simply said, we are going to take the Universal Declaration of Human Rights and the right of everyone to benefit. That's the actual wording, the right of everyone to benefit from scientific progress. In that same article in the Universal Declaration, that same article that talks about the right of everyone to benefit from scientific progress, you also have the right to the recognition of moral and cultural contributions. In other words, intellectual property. So it recognizes that innovation should and can serve humanity, but it also, and the citizens, but it also recognizes that scientists should be given proper attribution for their work. So GA4GH has a whole series of what I would call work streams. A lot of them I don't understand, highly technical, often like some of the biological and IT stuff we've seen here, on discovery, on data use, on clouds, and so on, APIs. And it has two transversal streams, foundational streams, one on ethics and one on data security. So what has GA4GH done to date? Well, it's, in the last 3 years, encouraged what we call driver projects to come to GA4GH with their issues, with their particular needs and say, or their particular contributions, this is what we're doing to promote global data sharing. And HCA is one of those driver projects. So there's about over 30 driver projects now. It's in over 50 countries. So it's getting quite big. And from the ethical point of view, we just started building what everyone wants first, which is the consent tools. So we looked at the different types of projects that could contribute. There's your definition. Lawyers always start with definitions. It's particularly the slides on GA4GH. Thank you very much, by the way. The foundational principles, this particular human rights framework, which allows us to be more universal and get over all these sort of, in Europe we can do this, in the United States we can't do that, and all that. In Japan we do this, and it moves it to a level that's already been ratified and recognized by the United Nations. You can see the little logo of the Human Cell Atlas down there, real-world driver projects, giving us real-world issues to solve. And then finally, coming to the consent tools. So this is the most difficult, believe it or not, step in addition to IT security issues and re-identifiability in the genomics world, and that is the whole issue of consent. And almost every workshop comes to this. So how can we simplify it? We looked at consents from over 85 different countries. We simplified them, we anonymized them, we created generic template clauses, and that you can then customize to your particular research project, be it population research, be it disease-specific research, be it pediatric research. Developmental tissues were particularly addressed in a document called Issues with Tissues, where we looked at the way 8 different countries dealt with different tissues. So this is what's up there right now if you're interested, and they have been approved all the way through the regulatory ethics workstream, all the way through to the board of the Global Alliance. Particularly to children, we have looked at pediatric issues. It's a particular field of interest to me from a political point of view, children being largely neglected till the 80s in terms of being included in research. So are there issues? We've not only created consent clauses, the ethics working group adopts the general position, it's not worldwide, but the general position that information coming out of genomic studies, if it's clinically actionable during childhood, should be communicated in the child's best interests so that the child can be treated, and we would hold irrespective of parental positions on this. So this is, you know, we don't not only do tools, there's also policy positions coming from the GA4GH. Now, suppose you get there and you have this sort of generic customizable consent tool, you also need to look at how can we, and we saw that this morning in some of the slides, how can we have a data use ontology where we can tag genomic data sets with both permissions and restrictions so that automatically through a IT system we can see what can be authorized for use and what's discoverable. And this is a data use called a data use ontology duo. And the challenges, of course, is to get the depositors then to deposit their data sets, tag the permissions and restrictions, and then be able to have access in, more rapidly. And that's why the more harmonized the consents are, the easier it is to do the duo, and the faster the data can be accessed and used. So there's 2 more slides, which I'll just, they can be disease specific. You can see some of the modifiers over there on the right. I don't have time to go into it, but the terms reflect disease, reflect categories, reflect age, permissions, restrictions, and what kind of approvals are necessary. that's not enough. Suppose you've got that, you've actually got yourself organized, tagged, and everything, and into a discoverable system. You still need to check the researcher who's asking for access. So what's been developed is something called a passport. It's an authentication process where you check that the institution actually recognizes this person as a legitimate, approved researcher, and then that person is then given a passport so they can go to the Access Committee and ask for data. And this is the Access Committee. It is being set up as well in HCA, as I mentioned. I prefer data access committees to handle data access requests. I know some countries send researchers back to the original ethics committees, but ethics committees are not constituted in a professional way for data access issues. They're more clinical trial oriented. They lack epidemiologists, they lack IT people, and so on. And so data access committees have the know-how, if I can say that, to properly provide governance over access requests. And again, GA4GH has developed a policy. So for each issue, consent, privacy, accountability, access, and so on, we have developed policies and tools that you can use in your country. So finally, that's my last slide. I'm having trouble with this slide. I did it maybe 2 weeks ago. And the reason I'm having trouble is yesterday in the session where there were the frogs and so on, there was also a bridge with a train on it. I don't know if you remember. And the speaker was talking about connectivity and plasticity and rigidity. And I thought, what's happening to my train here? The idea here was to say these are ethical provenance. This is how you can track consent, authentication, data access committees, and finally, data transfer as rapidly as possible. This might be one of those nice Japanese trains. And then you can track and go all around the world and find out what's happened. And then last night I was thinking, well, maybe it's the tracks that should have these names on them. And what actually are the little cars, or maybe the research projects seeking access. I don't know if any of you has an idea of how to fix this slide. I'd appreciate it. But that's the idea behind ethical provenance. Thank you.
Thank you again to everyone. I think all this ethical discussion and data is at the very core of the Human Cell Atlas. So a big clap for everyone again. And yeah, we will be back in 10 minutes.
So now we go. I'm not sure this one works. We're going to have a quick panel discussion.
Partha. So hello, everyone. We are now at the stage of some kind of like a future perspectives and a quick panel discussion, but maybe the panel could move a bit more to the center and make it a bit more equitable across the floor. So for those, I mean, I think, firstly, I would like to thank all of you for joining us at this meeting and for your active participation throughout the meeting, amazing contribution to the breakout sessions, and thank you very much once again. So I've got here, and you may have met all of these individuals over the meeting, but I thought it would be nice to just sort of like briefly mention. I'm Ms. Hanifa. I'm one of the organizers of this meeting. I'm also one of the coordinators of the Developmental Bionetwork for the Human Cell Atlas, and that's together with Stanley Nassen and Deanne Taylor. And I would like to take this opportunity to let everybody know that Alan Shadotal has now joined us as one of the coordinators. So that's fantastic news and great to be working together. And in addition to Alan, whom I've just mentioned, who is the main organizer of this meeting, we've also got Gary here from Toronto, who is also one of the co-organizers, and then Genevieve from Paris here, who's also one of the co-organizers, and Partha Majumder from India, whom you heard earlier. And this is just to sort of like have some discussion about our reflection of the meeting, the contents, where we think, you know, we should move forwards, the areas, challenges ahead. And while we discuss this, it'll be great if any of you who have comments to please sort of just put your hand up and we can pass a mic for you to participate further. So I'm going to kick off with the first sort of like discussion point question, which relates to, you know, the fact that we've got people from various expertise and domains here, and what would be the kind of future direction in terms of working together with the different societies, the different communities, particularly from the perspective of development, with the developmental biology community, with the pediatrics, with the clinicians within pediatric community. And I thought I'll ask that question for the reflection to Genevieve first.
Well, first, I'd like to say that it is really very important that we can bring together these different communities to look into the potential that can be really harnessed with approaches like the one that HCA has developed. So my feeling is at this point in time is that we are still in the process of the mapping, and so the aspect related to the underlying mechanism is really still a very important aspect that will help to refine the maps. And I really liked some of the comments that were made with respect to the use of disease as models of situation where you have perturbation. And so I'm just thinking that this is perhaps something that would be worthwhile exploring in more dimensions. So there was some doctors, but we may also develop that. The other aspect was also related to physics and parameters that we have not yet incorporated into our thinking. And so there's certainly here some thought to go deeper into how we can connect the information from mechanical properties of cells with the other features that we are looking into. And then another aspect is we've been at the level of the factors that are quite, we are at a cellular level, but with transcriptomic data, we haven't actually looked at parameters at the nuclear level in that much detail. And so I think this may be an interesting area to look into, especially with respect to shape and state. And so there's perhaps a lot more we could learn from the nuclear properties and the interconnection between chromatin and the nucleus. But this is sort of my own standpoint, but I would love to discuss a bit more these aspects as well in the future.
Thank you, Genevieve. My next sort of like panel discussion point is going to be about how we coordinate and also how we are representative globally. I mean, it's quite nice to think that there are many of us from different countries standing here, but there are also many initiatives. And my first question with regards to sort of coordination would be for Alan in terms of how you think we might be coordinating some of the developmental programs that are ongoing at, you know, to some extent at national level. So there are a few whole embryo projects ongoing in the UK, Sweden, and France, for example. You know, what are your thoughts on how we can kind of work together more seamlessly and synergistically?
I'm not sure it's working. Maybe, no, it's on. Okay. No. Yeah, I think one thing is clearly to meet more often. No, I mean, really, and to try to interact and try to also avoid like doing the same thing. So that's really important. So try to, I mean, people in Sweden they work on heart, in France they also work on heart development. There are people in Netherlands working on heart development, so it would be just nice that people work together to make sure that you don't just try to scoop each other, or that there's really a benefit, and there are different technologies that are used, so it's really important to really be coordinated. So we discussed yesterday with Olivier Pourquier, for instance. I don't think there's any network on muscle development. There's one on retina, but I think those networks, they should be like maybe more like officially or better organized, so to make sure that everybody is included. So I think there's a need to structure a little bit this, because like to make sure not like Olivier, you know, okay, is anybody working on muscles? And I told him, no, if there's no one, then you can maybe try to organize a network yourself. But maybe we should have some kind of guideline that we put together to try to identify specific organs, and people can lead some consortium and try to coordinate for every type. Like the heart was also an example, as I said, where I think there's a need to have some coordination between the different groups. So it's probably something we can propose.
Yeah, no, I think that's a great suggestion in terms of trying to see if there are people who are willing to sort of like be part of this coordinated effort. I mean, certainly, you know, there are lots of efforts ongoing, but also with the publication bundle, this is another way to coordinate it, and that will also ensure that, you know, everyone's work is going to be sort of published, you know, in as best a complementary fashion, but that's a good point. So the third question or discussion point, I'm going to direct it to Partha. We talked about coordination across ongoing projects, but one of the, you know, kind of spirit of the HCA has always been trying to be representative and inclusive. And that works very well, perhaps, for adult or postnatal tissues. But development has these challenges in terms of how do we ensure geographical representation, and I wondered what your thoughts are in terms of how we could actually increase participation from a broader range of researchers for human development.
Yeah, it's a challenging question. One is that, you know, in terms of the pediatrics network that's building, I think the challenges are not so many, but when it comes to embryos and those kinds of things, there's a lot of differences of viewpoints on handling embryos, when you can handle, and so on and so forth. And there is no harmonization of thought in different countries. Also, religious aspects are enmeshed with these kinds of things. So it's, I think, you know, handling of embryos and looking at development from that stage is going to be a major challenge. And I really don't have any major thoughts in terms of harmonization of, you know, the tissues. The data, once the tissues are collected, of course, the data harmonization may not be a problem. I really think that the leadership should go and talk to these various groups that are actually spearheading, you know, in the different countries, and see what the challenges are. And I don't think that we have a clear idea of the kind of challenges that we will face in order to garner those tissues and put it in, let's say, a global biobank. I mean, I'm not suggesting that there be a good global biobank, but, you know, conceptually that's what it is. So I think, you know, just this development efforts have just started, or these programs have just started. So I think we need to understand these various challenges and talk to our counterparts in various countries and understand what the challenges are, sort of make a listing of the challenges, and then see how we can overcome those.
Thank you, Partha. I want to kind of like direct Gary with perhaps two questions, actually. One is something that has come from these discussions in terms of, you know, collaborating, working together in a more efficient way, and therefore something that people might want to know if they don't. How do you join HCA? And also one of the biggest challenges of all of this is actually bioinformatics and computational. And I thought Gary would be the perfect person to sort of elaborate a little bit on those 2 points.
Yeah, it's a great question. Some people ask me at this meeting, you know, how do I join HCA? I guess my general thinking about this is that, you know, everybody in the world who is interested in single-cell genomics and mapping tissues, especially human tissues, and any kind of relationship, or even just interested in that, is already part of a community. You know, the HCA is trying to organize efforts that help this community. And so, for instance, the publication bundle is an example, or this meeting to bring people together, or connection with funders, to tell funders what's needed from the community. So all of this communication, convening, community building, frameworks that are put in place, the ethical working group, the diversity policies that are coming out. So these are helping the community. They're allowing, contributing to community growth, and anyone in the community is welcome to participate in any of those efforts. And so if you're interested, you just have to volunteer, and there's a online registry that you can sign up for that makes sure that you get emails, that you're informed about these things. Now, once you have this community, what is, I mentioned already a number of valuable aspects, just focused on the computational side, or even maybe just any kind of technology side. One of the most valuable aspects for me when I started participating was just learning from everybody, right? So as a computational biologist, I can share knowledge and code and other things, and we're also learning, and our collaborators are learning about how to best use single-cell genomics technologies and what's the latest technology. The talk by Ali is, amazing. How do I access that technology? And so that's one of the powers of the community, is all this information sharing to allow us to take our individual research projects and potentially take them to the next level with access to the best knowledge from around the world. So just from a computational point of view, again, and many computational biologists are participating, we're happy to answer questions and share, but it's a multi-way connection.
Thank you very much, Gary. We've had some of the reflection from the panel members here, and I, would like to open it to the audience if you wanted to add any comments relating to the points that have been raised, or any other points that you would like to raise. Okay.
So I want to say, maybe contrary to appearances, I'm not a frog chauvinist. I work with many different organisms, and I just want to share my general impression that people do not consider much model organisms in their work, and homology, tissue homology, cell type homology is not very much on people's minds. I think that we can do a better job of just compiling this information, making it accessible. Things like which organisms have certain tissues, and this organism has 3-camera heart versus 4-camera heart, and which kind of brain tissues are accessible, and also many resources which already exist for lesser model organisms. Mouse, I think, is very well integrated, maybe with human, but lesser model organisms could be just federated and today and made more relevant to Human Cell Atlas. I would be interested to speak to anybody about organizing this sort of effort.
Thank you very much. Yeah, that's an important area that we should definitely remember. Genevieve, did you have a feedback?
Just perhaps also along those lines, I thought it was actually very interesting when we heard the talk from Madeleine Lancaster showing the comparison between different species in terms of the size of the brain and certain properties, that indeed it can be really important to also have an evolutionary perspective on the question we're asking. So it could be, the use of model organism, but also other non-model organism. And so I think perhaps we need also to think a little broader, because we can access now to information from other species that could be very interesting and give us insight to better understand what we see in other in human, we tend to be human-centric. I don't know whether human is the most important thing, but the diversity is important.
Yes, there's a place for everyone, not just frogs, I suppose. So And I think all of those are important, and in fact, there was quite a lot of discussion with organoids, etc., and in terms of comparison that we'll take on. I'm conscious of the time, and I think, sorry, is that a sign? Oh, okay, sorry. I'm allowed to take one more question. Is there anybody else? Oh, okay. Sorry.
It's not really a question, more a suggestion in terms of the point you raised about getting access to tissue samples, especially embryonic samples, in more diverse places. Yeah. I think it would be really useful to have more community support in like understanding how to even go about setting that up. Like if you can share just even like the technical admin details of like who would be the first sort of organization you approach in order to ask whether we can get that sort of tissue donated. And I know like how it works in Europe may be different in Africa, but I guess there could at least be like, okay, you should go and talk to this sort of center, and maybe people will even have contacts in different countries to like help even places like South Africa to get involved, because I don't think there's anywhere collecting those sorts of tissues in that I know of in Africa, and I think it would be really important for to get advice to even just help those first steps to happen.
Hear me? Oh, okay, now. Yeah, that's a very important comment, and I think that would be a great use of the HCA network globally to find out more about contacts and how we might go about it, and also more amenable with the various platforms for analysis, and even with paraffin-embedded tissues that could open up huge opportunities moving forward. So thank you very much for that comment. Unless there are any other burning comments or questions, I will now leave Alain to kind of like give the closing remarks. And thank you very much for the panelists for their contribution.
So thanks to all of you. Whoops. This morning is a bit tough. One thing I wanted to do, because it was also one of the goals, is can people in the room that never attended any of the HCA meeting before raise their hand? Okay, see, it's nice. I knew it, kind of. No, it's really important. So I think that's a good objective. That was an objective for us, and it was also the idea was to bring also people from the developmental biology community to this meeting, and we need more. We need more interaction. That's partially something that we did, and I'm glad to see that people are working on like some weird models who came to the meeting. But I think we need more like interaction between data biologists and people from this community. But at least we're making progress, so it's nice. And again, try to talk to the other people and in your field, and try to tell them that it's important for them also to come here. They might learn some new technologies. They might also have good ideas. And also we need the ideas like, okay, what should you do now that you have those technologies? What are the key questions to address? And that's really something that would be important for us. So there's a lot of acknowledgement. So we want to thank you, of course, for attending it. I mean, it was great to see you in Paris and to meet many new people. We have also sponsors, of course, that allowed you to have this great food and the coffee breaks and everything. So thanks again to the sponsors, INSERM, Zucchi Initiative, Company of Biologists, CNRS, and Epigenesis. So we spent almost a year working on this program with a meeting every 2 or 3 weeks. So it was busy, but it was very well organized and I hope productive. So I want again to thank all the co-organizers, Geneviève, Gary, Muz, and Stan. I want to thank, and we want to thank all the record session leaders. I mean, who really, most of them, I mean, almost all of them, were answered immediately and say, yes, I'm going to come, I want to join the program. And then it was very nice to have them here. And again, to bring some new people to those discussions. So many of the leaders are listed here. Of course, we also want to thank Sarah as well, who's not here, but I see her here, who has really been the driving force for this effort since the beginning. On my side, I'd like to thank also people like Christine and Tracy that are in the back, who really were really like very helpful, did a lot of work. I mean, you haven't noticed her, but they were in the room there working on all the slides, and pushing us also a lot, trying to keep on time, and they did a really great job. And Samantha took all the notes together with Ellen as well. There are a lot of people that I won't have time to go through everything, but I want to send also people from my lab, like Amin, Raphael, and Emmerich, who run everywhere. Initially, they signed to just pass the mics, and then they ended up in the kitchen, like doing with all the trash and all the things. So I apologize. I didn't know it would be like that. Next time we do better. And also I want to thank people here at the university also who were very useful, and Matura in particular. I don't know if he's here today, but he helped a lot on the weekend, on a Monday, to really like open and organize everything. And I guess that's it. So one key thing, so it was already mentioned with publication bundles. And I think there's another slide. Yeah. So all of you, I mean, as I say, you're new here, so you're not, maybe many of you are a member of HCA. It doesn't cost anything. You just need to click, and then you will receive some newsletter. You will receive announcement about meetings. So join HCA and try to build a larger community and talk about it to your colleagues. And I hope you enjoyed the meeting. And I think that's the last slide, if I'm right. One more. Thank you. The survey. Okay. So there will be a survey, I guess. So I guess they want us to do the survey, and so it's also important, I think, for future meeting also to get feedback, as it was mentioned before. We know it's I mean, a bit always like goes in all direction. The breakout session also, we need to know whether you like them, and if there's some topics that you think are essential that were not discussed. So for the next meeting, then we can add some new breakout sections and then really address other questions that we didn't address this time, or some question we addressed that you think there was not enough time, maybe there's still more to say about it. And so let us know. It's very important for future meetings. I guess that time that was the last one. Thanks. Okay. Thank you. So, and have a good trip back.